Nosemosis is a condition in colonies that's caused by one of three different species of Nosema disease. Now, Nosema is a genus of organisms. They happen to be single-celled fungal organisms. In this case, the three species I'm talking about live in the midgut of the honeybee. So what are those three species? There's Nosema apis, Nosema ceranae, and Nosema pneumoniae. Nosema pneumoniae was very recently discovered, so there's not quite as much known about it. So the rest of this presentation, I'm focusing primarily on Nosema apis and Nosema ceranae. For that matter, if we want to get very technical, here in the U.S. as an example, while we've had Nosema apis historically for over a century, it's largely been replaced by Nosema ceranae. So most research projects that I see today are on Nosema ceranae because it seems to now be the more prevalent of the species of Nosema infecting bees. So with all of that said, I'll talk a little bit first about Nosema apis because it's the one that we have known so much about because it's been known for decades and decades and decades. Again, it's just been replaced largely by Nosema ceranae. So with Nosema apis, it infects the midgut of bees and it leads to a few different clinical manifestations. One of those is just being dwindling colonies. Colonies that look like they have the resources they need, life should otherwise be good, but the bees aren't thriving. The colony is just dwindling in population. Nosema disease tends to be a bigger issue coming out of winter because you've had all of these bees put together in this really tight area, transmitting the disease to one another. So Nosema apis is often associated with fecal streaking on the front of the hive. The idea is that these heavily infected bees, when they finally are able to leave the nest after winter subsides, they'll defecate the moment they leave the nest and streak feces on the hive. It's never been conclusively shown to be caused by Nosema, but it's often believed that fecal streaking is associated with Nosema apis. You also can get a condition called K-wing. Remember, bees have two wings on both sides of their bodies, a large wing called the forewing and a smaller wing called the hind wing. Those two wings, when the bee is at rest, typically are folded nicely beside the body. K-wing is a condition where those two wings kind of become unhinged, so you look like the letter K. So bees will display K-wing. They often might wander away from the hive. You'll see them on the ground outside of the nest just crawling away or crawling up blades of grass. The problem with these conditions, K-wing, this wandering away from the hive, crawling up blades of grass, is other things in honeybee colonies can cause these as well. So with Nosema apis, people were typically looking for that. In fecal streaking, they were looking for the time of the year, again, coming out of winter, and all of those things suggested that it might be worth a closer look to see if colonies have Nosema apis. But flashback to around 2006, 2007, Nosema ceranae was discovered. And when people started looking, it turned out to be the dominant Nosema in most of the samples they looked for here in the U.S. And I started to hear those similar trends all around the world. So our default Nosema, when we think we see Nosema in our colonies, it's almost always Nosema ceranae. And again, I start to hear that from my colleagues and from beekeepers around the world. So what does it do? Well, it's a little tricky. It's not necessarily associated with fecal streaking. It's not necessarily associated with coming out of winter and dwindling bee populations. What it appears to do is it appears to just shorten bee lifespan. There's been lots of research projects on this disease where people will infect bees and get a response. They'll have shortened lifespans in cup studies, cage studies, maybe even colony studies. There's been people who've done similar studies and found no response. But Nosema ceranae, generally speaking, is believed to shorten bee lifespan and encourage other stressors to potentially take over the nest. You can get dwindling populations of bees, etc. Just think about it this way. Both Nosemas seem to make colonies that should otherwise be growing and going, make those colonies dwindling when resources are supposed to be good out there. So how is Nosema spread in the hive? Well, again, if it goes into the midgut of bees, it can be spread through trophallaxis, bees feeding one another. If it's associated with bee dysentery, which Nosema apis was believed to be associated with bee dysentery, then bees simply defecating in the nest, outside of the nest, etc., can cause the spread of that disease as other bees clean it up. There's even been pretty good evidence that bee collection of water can be a really good way to spread the disease. So if you think about it, you can think about it almost like dysentery that's spread everywhere, and then bees just pick it up when they're going back to feed one another or feed themselves. Once it gets into the midgut, a single Nosema cell will penetrate one of the cells that line the wall of the midgut. Those cells are called epithelial cells. So that Nosema spore will inject genetic material into that cell. More individuals will be made, and then that cell will rupture, releasing more Nosema spores that then go on to infect other cells that line the midgut of the bee's gut. And if you think about that, all of these cells are rupturing, all of these cells are infected. What you do is you get a nutritionally compromised bees. The epithelial cells that line the midguts are those cells that start that process of handling nutrients that come into the bee's midgut through the consumption and then further the digestion of food. So if you've got all of these cells that are compromised and rupturing, what you get is a bee that looks nutritionally stressed. She's being outcompeted for the food that she's taking into her body by the mechanisms of Nosema designed to make more Nosema and therefore rupture the cells in the nest. So when you think then about Nosema control, one of the best ways to address it is nutritionally. Now, you're going to hear me kind of be very non-committal with a lot of my comments about Nosema, and that's because we're still learning a lot more about this more prevalent species now, Nosema ceranae. So there's some evidence that Nosema spores, for example, might go up in protein-rich diets. So when pollen is coming into the nest, or maybe when pollen subs are fed to colonies, you can get more Nosema in the guts of bees. On the other hand, these things still seem to help bees pull through the Nosema infection. So that's why one recommendation is feeding. Just give them more nutrients to try to help the bees just overcome it on their own. Another option for you with regard to Nosema is using Fumagilin. Fumagilin is the only treatment that's labeled for use in honeybee colonies. Now, why am I a little bit non-committal here? Well, there are some studies that have shown Fumagilin work quite well and reduce Nosema infections in bees, and other studies that show feeding bees Fumagilin did nothing at all to the Nosema population. So really, if you believe you have Nosema in your colonies, you're going to have to either try this or not try this, or talk to other beekeepers in your area to see what their experience is. How can you know definitively that you have it? Well, the only way really to know is to take samples of bees and perform a dissection to look for those spores in the midgut, or you can send those samples off to a diagnostic lab that will do dissection or use molecular technologies to identify it. But if you have it, you're kind of stuck in those two areas. Try to feed the bees out of it, or consider using Fumagilin according to the label. Unfortunately, neither of these are perfect options that work all of the time. The good news is, if bees otherwise have their varroa pressures under control, have ample nutrition, their queens laying lots of eggs, and life is otherwise good, most colonies can kind of pull through it. It will compromise their health, their integrity, their productivity, but most colonies can pull through it. In fact, just outside a door over here, we keep our university's apiary. If I were to go into those colonies, I would say the vast majority of those colonies have Nosema in the colonies, but the colonies are functioning well and otherwise doing good. So it's not always necessarily something that you have to deal with. You can hope your bees make it through if there's nectar-rich nectar flow zone or pollen-rich pollen flow zone. But if not, you might consider defaulting to Fumagilin and seeing how that works for you. Nosemosis is this disease that many colonies around the world are going to have. I know that I see it in bees nearly every time we dissect for it here at the lab. We have mixed response to trying to do something about it. There are a lot of new technologies out there, people studying propolis or some of these other hive supplements that are advertised in the equipment catalogs to help against Nosema. Just keep in mind, this is an ever-evolving science. Recognizing the infection and then taking steps to deal with it will ensure that your bees are healthy and productive well into the future.